Welcome back to the Deep Dive. Today, we're not just reading a medical paper. Honestly, we're looking at what feels like a tactical map for a siege.
That's a good way to put it.
We're tackling the black box of childhood brain cancer, specifically medulloblastoma. But we aren't just looking at the tragedy of it. We are looking at a brand new, massive study. This is from 2026 by Bernardi and colleagues that effectively hands us a blueprint to a secret weakness.
And blueprint is exactly the right word. For the last 20 years, treating medulloblastoma has felt like trying to navigate a city with blurry, hand-drawn sketches.
Right. You know the big neighborhoods, but that's about it.
Exactly. You don't know the streets. You certainly don't know how the traffic flows. This is like switching to 4K satellite imagery with real-time traffic data.
I love that imagery. We're talking about a study titled Multiomic Integration Reveals Tumoral Heterogeneity of Lipid Dependence Within Lethal Group 3 Medulloblastoma.
It's a mouthful.
It is a mouthful. But by the end of this, you're going to understand exactly why that title should give parents and oncologists a lot of hope. So let's set the stage. What exactly is the problem we're trying to solve here?
The problem is heterogeneity. Medulloblastoma is the most common malignant brain tumor in kids. And, historically, we've put patients into these 4 big buckets or subgroups: WNT, SHH, that's Sonic Hedgehog Group 3, and Group 4.
But the buckets are leaking.
They're leaking badly. You could have 2 children diagnosed with a Group 3 tumor, give them the exact same chemo, the exact same radiation, and one survives while the other succumbs within a year.
Which tells you right there, those 2 tumors weren't actually the same thing, even if they look similar under a basic microscope.
Exactly. And that is where this paper it begins. The researchers didn't just look at the DNA. They used what's called a multiomics approach on nearly 400 tumor samples.
Okay, let's pause on that. Multiomics. I want to take a second here because this is the whole backbone of the discovery, right? Omics just means the whole set of something.
Right. And they looked at 5 whole sets, 5 layers of biology.
Okay, let's walk through them. Layer 1.
Layer 1 is the methylome. You can think of this as the packaging of the DNA. It decides which files are even accessible.
Like the software that decides which programs you're allowed to open.
Perfect. Layer 2 is the transcriptome. That's the RNA, the actual instructions being copied from those accessible files.
Okay.
Layer 3, the proteome. These are the actual worker proteins that get built from those instructions.
The machines on the factory floor?
Then layer 4, the phosphoproteome. This gets granular. These are the on-off switches for those very same worker proteins.
So not just the machine, but whether the machine is running or not.
Precisely. And then layer 5, which becomes the absolute star of our show today, is the metabolome.
Which is the fuel and the exhaust from the factory, right?
That's it. And it's so important to get this distinction. The genome, the DNA, tells you what the cell might do. The transcriptome tells you what it's planning to do. But the metabolome...
It's actually happening right now.
Yes. It's the difference between reading a restaurant's menu, that's the genome, and analyzing the food that is actually sitting on the diner's plates. That's the metabolome.
That is a perfect analogy. You can have a salad on the menu, but if everyone's eating burgers, the menu doesn't tell the real story.
Exactly. So they take these 5 massive layers of data and they feed them into an algorithm called MOFA.
MOFA
stands for multiomics factor analysis. It's a computational tool that's just designed to find patterns across all these layers that a human brain, you just couldn't see it.
And MOFA didn't just find a pattern. It blew up the map.
It totally did. It revealed that those 4 standard subgroups, they're actually made of 9 distinct proteomic subtypes. It's like realizing what you thought was one big country is actually 3 smaller countries with different languages, different economies.
And this is where we meet our villain for today. The algorithm splits group 3, the really lethal 1 into 3 specific subtypes: G3-PAPA, G3PB, and G3PP.
And we are hunting G3PAPA.
G3-PAPA. Why that one?
It is the deadliest. It has the absolute worst prognosis. And genetically, we know it's driven by high levels of an oncogene called MYC.
MYC. We've talked about MYC before.
Yeah.
It's like a master regulator screaming, grow, grow, grow at the cell.
Right. And usually that growth is fueled by sugar, glucose, the famous Warburg effect.
Yeah, that's Cancer 101. Cancer loves sugar.
That's what we thought. But here's the surprise twist. When they looked at the metabolome, the actual food on the plate G3-PEP wasn't eating sugar. It was stuffed, just overloaded with lipids. Fats, specifically triacylglycerols.
So hold on. While all these other tumors are running on a sugar rush, this one, the most lethal subtype, is running a diesel engine. It's obsessed with fat.
That is exactly it. It has completely rewired its entire metabolism to be a fat-burning, fat-storing machine. Yeah. And that discovery, I mean, that changes everything about how we fight it.
Okay, this feels like a major pivot point.
Yeah.
Before we get into how it's do this, let's do our first recap. We're about 5 minutes in. Let's lock these concepts in.
Good idea.
All right, quick recap. Point 1. Medulloblastoma isn't just 4 groups. This multiomic study shows it's at least 9 distinct subtypes, a much higher resolution map. Point 2. The deadliest of these is a subtype called G3PA, and it's driven by the MYC gene.
Right. The accelerator pedal is floored.
And point 3, the big one. Unlike most cancers that gorge on sugar, G3PA is a metabolic outlier. It is overloaded with lipids. It's a fat-fueled tumor. How's that?
Perfect. We've identified the enemy and its fuel source. Now we have to understand the engine.
So we have a fat tumor.
Right.
The next logical question the researchers asked, and they used a tool called Cosmos to do it, is how? Yeah. How does a gene like MYC tell a cell to, fill up with grease?
So Cosmos is a causal analysis tool. It's designed to connect the dots from the gene to the metabolite. And it showed that MYC is acting like a factory foreman.
Okay.
It's forcing the cell to start building fat from scratch. The process is called de novo lipogenesis.
Making new fat.
Yes. And MYC does this by cranking up 2 key enzymes. The first is FASN, that's fatty acid synthase. Think of FASN as the assembly line robot that builds the raw fatty acid chains.
The basic building blocks.
Exactly. The second enzyme is SED, or steryl-CoA desaturase. SED is like the refiner. It takes those raw chains and processes them into more specialized unsaturated fats, like oleic acid.
So if I'm visualizing a factory floor, MYC is the foreman yelling, faster! FASN is the machine churning out raw material, and SED is refining it into high-grade fuel.
You've got it. That's the pathway.
So here comes the obvious trap. And I say trap because biology is never this simple. Thought would be, great, let's just shut down the factory. Block FASN or SED with drugs, the tumor can't make fat, and it starves. Simple.
That is exactly what logic would dictate. And you know what? In a petri dish and a nice controlled lab environment, it actually works. You block FASN, the G3PA cells die off. But when they moved this into live mouse models, specifically what we call patient-derived xenografts, where you have the actual human tumor growing in the mouse, it failed completely.
The tumors kept growing.
Just kept on growing.
Okay, that's the plot twist moment.
Yeah.
The factory was closed, the FASN machine was broken. So where were they getting the fat from?
They ordered takeout.
You're kidding.
I'm not. The tumor showed this incredible plasticity. When its internal factory was shut down, the tumor cells just opened their doors and started scavenging fat from the surrounding environment.
From the blood, from the brain tissue.
Wherever they could get it, it's called exogenous uptake.
That is terrifyingly clever.
Yeah.
So you block the synthesis pathway and the tumor just says, fine, I'll just eat what's floating by.
Exactly. And this is why so many metabolic drugs fail in the clinic. We underestimate the escape mechanisms that tumors have. We think they're these rigid, broken machines, but they are master survivors.
So we can't starve it by stopping production. Just steals food instead. But the researchers didn't give up. They noticed something else about all this fat, right? It wasn't just like floating around loose inside the cell.
Correct. And this leads to the real aha moment of the paper. They realized that high levels of free fatty acids, it doesn't matter if they're made inside or stolen from outside, are actually incredibly toxic to cells.
Lipotoxicity.
Lipotoxicity, it's a huge problem. Think of free fatty acids like gasoline. If you have gasoline safely stored in a tank, it's fuel. If you have gasoline spilling all over the floor of your living room.
It's a massive hazard. It's flammable. It's corrosive.
Exactly. It dissolves membranes. It disrupts signals. One spark, the whole house burns down. The cell is the same way.
So the tumor is making and eating all this dangerous fuel. It needs a safe place to put it.
It has to. It needs to barrel the fuel. And it does this by storing the fats in these little organelles called lipid droplets.
Lipid droplets, okay.
And the worker, the enzyme responsible for taking that dangerous free fat and packaging it safely into these droplets, is an enzyme called DGAT1.
DGAT1. Okay, everyone listening, remember this name. DGAT1. This is the barrel maker.
That's the one. DGAT1 converts those volatile fatty acids into something called triacylglycerols, which are neutral. They're safe to store. It's literally like taking radioactive waste and sealing it in lead-lined drums.
So if it's in the drum, the lipid droplet, it's safe fuel. If it's loose, it destroys the factory.
That's the paradox. And the paper found that MYC doesn't just tell the cell to make fat. It forces the cell to store it properly using this DGAT1 enzyme.
Wow. But it gets even more elegant, right? I mean, the visual from the microscopy.
Oh, this is the best part. They used electron microscopy. We're talking super high-powered imaging, and they looked to see where these lipid droplets were physically located in the cell.
And they weren't just floating around randomly?
Not at all. They were physically touching the mitochondria.
The power plants of the cell.
Yes. It's a specific structure called a mitochondria ER contact site. The lipid droplets were essentially docked onto the mitochondria, spoon-feeding them fuel directly for energy production. It's an unbelievably efficient system.
It's like having a fuel tanker truck parked with a hose connected directly into your generator. No spillage, no waste.
Exactly. It lets the tumor run its metabolism at this incredibly high rate without blowing itself up with toxic byproducts.
Okay, this is the perfect time for our second recap because we're about to pivot from the problem to the solution.
Let's do it.
Recap number 2. 1. We tried blocking fat creation using FASN inhibitors, but the tumor outsmarted us by scavenging fat from the environment, the takeout method.
Right. Escape mechanism number 1.
2. All that fat is actually dangerous. It's toxic unless it's stored safely.
The gasoline on the floor problem.
3. The key enzyme, DG81, is the worker that packs this dangerous fat into safe lipid droplets. It makes the barrels. And 4, these droplets act like batteries, physically attaching to the mitochondria to directly power the cancer cell. Does that cover the mechanism?
Spot on. The tumor has built a high-performance engine, but it absolutely requires a very specific fuel storage system to keep from exploding.
So we attack the storage system.
Yes.
We attack DG81.
Precisely. The researchers hypothesized if you can't stop the tumor from getting the fat, maybe you can stop it from putting it in the barrels.
Take away barrels.
And if you inhibit DGA21, you create what they call a double whammy catastrophe for the cancer cell.
I like double whammy. Walk us through it. What's the first hit when DGA1 is blocked?
The first hit is starvation. Simple as that. Because the lipid droplets disappear, the mitochondria lose that direct fuel line we just talked about. Energy production it crashes, the lights go out.
Okay, so the engine stalls. What's the second hit? The second part of the whammy.
The second hit is poisoning. And this is the violent part. Remember, the tumor is still making and eating fat because MYC is still screaming, "Gah!" But now there are no barrels to put it in.
The gasoline is spilling all over the floor.
Everywhere. These free fatty acids, especially the polyunsaturated ones, they start building up in the cytoplasm, and they undergo a process called lipid peroxidation.
Which basically means?
They go rancid, they oxidize, they fall apart. And this triggers a very specific iron-dependent type of cell death called ferroptosis. The cell effectively rusts from the inside out and dies.
That is brutal. And did it work?
Extremely well. When they tested DGA1 inhibitors in those same mouse models, the ones where the FASN drugs failed so spectacularly, the results were significant. It extended survival dramatically. It turns the tumor's own gluttony against it.
I love the irony there. You take away its stomach, its storage capacity, and the very food it craves becomes a poison.
And it bypasses the escape route. That's the key. It doesn't matter if the fat comes from the internal factory or from the takeout scavenging. If you can't store it, it kills you.
This feels like such a monumental shift in thinking. We usually try to target the driver, right? We see MYC, and the goal is always turn off MYC. But MYC is notoriously, undruggable.
It is. It's like trying to grab a wet bar of soap. But this approach is different. It's targeting a metabolic addiction. It's saying, okay, we can't stop the driver, MYC, but we know this driver drives a car that requires a very specific type of fuel maintenance.
If we disrupt the maintenance, the car crashes. Doesn't matter who's driving.
Exactly.
So let's zoom out a little. Is this mechanism, this MYC-DG81 connection, unique to medulloblastoma? Because I feel like I've heard about MYC and lipid droplets in other cancers.
You have, but context is everything in biology. It's so important. Take, for example, renal cell carcinoma kidney cancer.
Yeah.
In that cancer, if you block MYC, you actually see an increase in lipid droplets.
Wait, the complete opposite.
The complete opposite. But here, in group 3 medulloblastoma, MYC drives their formation. It's a fundamental difference. And it just highlights why we can't copy-paste treatments from one cancer to another. You need this level of deep multiomic mapping to know which way the traffic is flowing in this specific city.
Okay, so the big question, what's standing in the way of this becoming a treatment for a child in a hospital tomorrow? I mean, a parent listening to this is going to want to know.
Yeah, and the biggest hurdle is the fortress wall. It's the blood-brain barrier, the BBB. It's designed to protect our brain from toxins, which is great, but it also keeps out most drugs.
Right.
The DGAT1 inhibitors used in the study worked in mice, but the next great challenge for medicinal chemists is to design one that we know can cross that barrier efficiently and safely in humans.
But the paper also gave us a tool we can use right now, didn't it? Something about biomarkers.
Yes, and this is immediate good news. Because we now know this mechanism relies on lipid droplets, a pathologist can look at a biopsy sample under a microscope. If they see a tumor just teeming with lipid droplets,
They know what they're dealing with.
they can say, this is a G3P tumor. This patient's cancer relies on DGAT1. It takes the guesswork out of it. It moves us from, let's just try the standard protocol, to, we know exactly what this tumor's weakness is.
True precision medicine.
It's the path forward.
We have covered a massive amount of ground today. We went from a black box of confusion to a 9-subtype map. We met the villain, G3P. We uncovered its deep addiction to fat. We saw how it cleverly escaped standard treatments.
By ordering takeout.
And finally, we found its Achilles heel. Not the fat production, but the fat storage enzyme, DGAT1.
It really is a masterclass in how modern technology, multiomics, and AI can unravel these biological mysteries that they've stumped us for decades.
Before we sign off, I want to leave our listeners with a final thought to mull over. We tend to think of cancer as a genetic disease, broken DNA, mutated codes. But this paper shows it's just as much a metabolic disease.
Absolutely. The G3 tumor is an engine running way too hot, and its fuel system is its weakness. We're moving from hunting for mutations to hunting for addictions.
So here's the question for you to think about. If a cancer cell can rewire its entire metabolism to survive, literally changing from a sugar burner to a fat burner to escape death, what other escape mechanisms are hiding in the data of other, incurable diseases, just waiting for a multiomics map to reveal them?
That's the question that keeps scientists up at night.
Thanks for listening to this deep dive. Stay curious, keep learning, and we'll see you next time.